Well, I think we have always viewed pharmacogenomics as a potentially important tool, but we view it certainly as people who practice medicine as one of many tools. And I think there are situations where pharmacogenomics is going to be completely meaningless. It will have nothing to do with some drugs. And there are situations where it will be very, very important. So for all of us, it's very hard to know which of these two extremes we're dealing with. So I want to try and address that by talking about clinical therapeutics in general. And this is a way of getting at how we can all follow in a practical way progress in clinical pharmacogenomics. And I think even people who've been in this for a long, long time, like me, are challenged by this particular thing. How much of our response to an individual medicine is genetic and how much is not, and how can we figure that out? And however long we've been in it and however much we know about it, we constantly make idiots and fools of ourselves by coming up and saying it's one when it's the other. And I think that falls on both sides of the line. We say things absolutely not genetic, has nothing to do with any genetic thing whatsoever, and is completely wrong. We do the opposite also. We say it's a big, strong genetic component, and it turns out not to be. So we have a history, unfortunately, of many mistakes, as is true in, I guess, all medicine and all science, but here we go. So it would be nice if when each of us took an individual medicine for an individual condition, we all had the same response. But of course, the whole discipline of clinical pharmacology is based on the fact that that's not true. But it's also should be based on the fact that we have multiple tools already available. So I think a question one always has to ask, if you have these tools, the ones in front of you here, the medication history of an individual patient, and I'll talk a little bit about that later, because we believe that the way we take medication histories as pharmacists, as nurses, and as physicians, to put it politely, sucks. We're very bad at it. We ask what medications are you taking, what allergies do you have, that's it. And really we need a lot more information than that, and we should do it a lot more carefully, and a better way of doing it is using a separate paradigm, which we call the avoid algorithm, avoid mistakes. The clinical effect of the drug varies from one person to another, but often we have some sense of that when you're talking to a patient already. You have some sense of whether they were nauseated or whether they felt better or not. So you can't ignore that when you're thinking about a genetic. Age matters to some things. Gender, I think, is way oversold personally. I think there are some things for which gender is important, but not many. Self-described ethnicity is recognized to be important, for example, in the response to hypertension, just as one small example. And obviously renal and hepatic function in the situations where we know them. Here's the avoid algorithm, and this was first published by our group more than 10 years ago now. So the idea here is you ask more than just what medication you're taking, what allergies you have. You have to ask the allergy question, but a better way of asking it is, are there any medicines we should not give you for any reason? It's just a habit you have to get into. And that gives you a lot more information than asking what allergies you have, and you end up with a lot more valuable idea of what the patient responds to. And as I'll indicate lower down, you can expand this later. You have nowadays to ask about vitamins and herbs. This is as big an industry as the pharmaceutical industry. I'll say it again. It's as big an industry as the pharmaceutical industry in any area of medicine you care to choose. And because so many prescription medicines prescribed by the normal healthcare system we live in have so many side effects that are not well described by our system, half the world in the United States and half the world in the world has divorced us and lives in this world of alternative medicine, which we're finding as we delve into it has all kinds of valuable things and some less valuable things. So it's valuable where possible to ask about this, and we could spend a whole time talking about it, but the best people to find out about this are generally not the people wearing the big white coats and who have the gray hair. The best people to ask these questions are usually the administrative assistants and secretaries sitting at the front, who are much less threatening to patients. Old drugs is something that's really actually important in pharmacogenomics, because some people keep medications in their system a lot longer time than others. And it's a very, very useful and interesting question to ask somebody: did you stop taking anything in the last 2 weeks? Did you stop taking anything in the last 2 weeks? And you can get very, very useful information out of that, and that should be part of a good medication history. You need a resource for drug interactions. That's not what we're going to talk about today. A resource for thinking about dependence, which we think is just opiates and other drugs of abuse, and that's a gross understatement. There are many, many medications for which patients should be counseled carefully about stopping. And last is Mendel, or your mother, or whatever you want to call the M. But I've been doing this all my practice life, which is now many, many years, asking people if they know, if their parents, sisters, brothers, their direct family relatives have any unusual response to medications. And I think I have yet to ask a patient who doesn't have some answer to that question, which is an amazing thing to me. I'm always amazed by the answer, and it's a good thing to do. Now, not all pharmacogenomic information is valuable. And this is just a cartoon to describe how little of it is actually valuable. If this bar here represents the whole human genome, which of course is, the numbers matter here a little bit, 3 billion nucleotides, okay? And of these, we have, among the single nucleotide polymorphisms that Dr. Decker described, somewhere in that ballpark of 10 million, maybe 20 million, depending on how we define it. Let me get these numbers again. 3 billion, and you have about 10 million SNPs. On the best, fanciest, high-tech technologies we have to screen at once, we have maybe 2 million plus SNPs that are interrogated on the Illumina and Affymetrix arrays. So they're a minority of the total. We try and make them represent more by using haplotypic approaches and others. But we have to remember, when we're doing any kind of rapid genome-wide screening, it's a tiny minority of the total. And that's why it becomes tremendously important later to think about next generation sequencing and the kind of things that Brian Schneider and others are going to talk about later on. Some of these are non-synonymous, meaning they change the protein sequence as well as the DNA sequence. DNA is degenerate, the genetic code is degenerate. I guess this is now elementary school stuff, but and some of them change activity of drugs in vitro, some change pharmacokinetics, some will change drug response, a tiny number, and a minute number change clinical outcome. And what I always say at the top here is an even smaller number change clinical outcome in an economically valuable way. By that I mean that we're able to generate a test that actually makes money for somebody. We live in the United States, and we're a bunch of lousy capitalists, but at root, if the test can't be sustained, and there's no business model that will allow it to be sustained, if it can't operate in this economy, we might as well not have generated it in the first place. And sometimes you can tell really early on, because of issues related to intellectual property or others, that it's going to be really hard to do that. So it's not an irrelevant thing to think about very early on. We spent a huge amount of time and money doing this, as Brian mentioned, several $1 billion in 13 years. And now really the challenge for all of us, I think, who practice medicine at whatever level, in clinical labs, as nurses, pharmacists, and as physicians, is to deliberately look for outliers. We're used to throwing out outliers and saying that's a weird response. Something went wrong in the lab, or something went wrong in the way I measured the blood pressure, or something went wrong in the way I assessed the rash, something like that. These are the people, from a research point of view, that are most interesting to us, the unusual weirdos, the people who don't have the normal response, or the data point you want to rub out is the most interesting. the most important one to go after. And that's what we're generating now, and we're interested in generating more data by going from the top here, from the unusual response down, because we have so much of a map now of the human genome. We can go in the opposite direction, find the weirdo response and go down and look hard and see what it is. Now, there's been a lot of progress. Brian mentioned some of this, and these are examples of commercially available pharmacogenetic tests. And this is just a slide. I don't want everybody to remember it, and you're not going to be tested on it. But it's to make the point that there are a lot of drugs that we use a lot. Orphan clopidogrel codeine is actually close to the most widely prescribed drug in the country. If you look at the largest number of prescriptions in any year over the last 10 years prescribed in the United States, and these are numbers in the hundreds of millions of prescriptions. Hundreds of millions of prescriptions. Always the opiates like codeine, oxycodone, and hydrocodone are right up at the top there. So this doesn't matter to a few people. This matters to a huge number of people and right the way across the whole of medical practice. There's very, very few physicians outside radiologists and pathologists who don't deal with anything living that don't treat pain somehow. You can tell I'm an internist, can't you? Tricycling antidepressants we don't use a lot in this country. They're used a lot more in Asia, Europe, Australia, the rest of the world actually, but they have strong pharmacogenetic effects. Brian mentioned one of the statins, but there are others obviously. HIV medications and these seizure medications here, which have nasty, nasty skin reactions that can be almost completely prevented by doing this genotyping. And a number of anti-cancer drugs, but in total, if you look at this, and I've been doing this a few years, cardiovascular things are a little represented. Neuropsychiatric things are less represented, and cancer is kind of in between. Reflecting, I think, the fact that we have a much better understanding of what's going on in cardiology at a mechanistic level. The brain is the last frontier, and I guess that's why this building exists. We have much, much less idea of that, and cancer is somewhere in between. So this is what I really want to spend time on. Which of these things matter at all? Which should we really care about, and which are just meaningless? Which should we just ignore? So I'm going to try and provide you here with a rubric, a way of going about helping decide which ones matter and which ones are a complete waste of time. And this is one way that I think is fairly intuitive. It's just a cartoon, but it's fairly obvious to people. So on the y-axis here, we have the real clinical value to you of a pharmacogenetic test, to someone prescribing a drug or to the patient. The real value, and by that I mean its value in terms of improving therapy and avoiding adverse reactions. And on the x-axis is our current ability to predict that response. You might say, I don't have any ability to predict response, but that's not true. It's nearly always the case that we have some sense of whether the patient's likely to respond. And one example I've put in here is, it's a bit fuzzy here, but is beta blockade with a beta receptor, because we can measure the pulse easily. Blood pressure medications are another example. We can measure pretty quickly, within days, whether blood pressure medication is working. So if you can tell by measuring the pulse with a beta blocker, or you can tell the blood pressure goes down with hydrochlorothiazide, why do you need some fancy test? And probably the answer is you don't. You know if it's working or not. You might not be able to predict the side effects, but you can predict the effects. And if you have a way of knowing the effect quickly, why on earth do you need some fancy pharmacogenetic expensive test? The answer is you don't. Now here's a more sophisticated example. One of the poster children in our field is this drug, azathioprine. It's a poster child, not because anybody knows what it does, but in fact, you probably get a Nobel Prize for understanding what it does. But it's metabolized by this enzyme called TPMT, thiopurine methyltransferase, and if that is not present, the patients are vulnerable to granulocytopenia, bad granulocytopenia, really bad, nasty, life-threatening granulocytopenia, and as a result can get life-threatening infections, ends up in ICUs and on antibiotics, and become expensive people. Granulocytopenia, of course, is low white counts, and so people have argued that, okay, if I can measure the white count, which generally is measured frequently in the patients put on azathioprine, why do I need some pharmacogenetic test? So this isn't a clinical test done by physical exam like measuring pulse or blood pressure, but it's a laboratory test, and it's widely used, like the INR, for warfarin. And white blood cell count is just like available easily. It's cheap. It's something physicians understand. It's not something complicated. We all can easily get it. So when the FDA panels first looked at this test, the pediatric oncologists and gastroenterologists on those panels said, I don't really need this test. I got the white count. And that's right, I think, except if there's something else involved than the immediate side effect. If it's not just granulocytopenia we care about. You can measure that with a white count. But if there's secondary cancers later down the road, many, many years later, that are actually predicted by the exposure to azathioprine predicted by TPMT, then there's something there. So it's a different thing. But for the white count and granulocytopenia, I'm putting it down here. It's not of massive clinical value because we have the white count. Now, antidepressants are a different ball of wax altogether. We're really bad at this, knowing which antidepressant to use on an individual patient first, second, or third. And there have been really big, randomized, expensive trials conducted over the last 5 or 6 years that just emphasize the fact that we're really bad at it. It takes on average 1.5 years to 2 years to get an antidepressant right in the people we get it right in, but there's a wide standard deviation on that number. And the morbidity incurred during that period of time, if you're taking ineffective antidepressant, the vulnerability to bad, not only to bad side effects, but also to worse depression and suicide is a huge public health issue. And I think we horribly need this, but we're a long way in terms of actually having clinical tests that can predict the efficacy of antidepressants at the moment. We need a lot more work in that area. And cancer chemotherapy and cancer therapy in general are probably right at the top because of course, you don't know a cancer drug isn't working very often until the cancer comes back, and then it's too late. So we have a very high need for this, and the bar for using these is much, much lower in some senses because we have so few abilities to predict responses to antidepressants and cancer therapy. This is really just an introduction to have in your packet for the large number of possible test platforms that there are now. And this is only increasing at the moment. I think it's probably going to consolidate. If I try and think 10, 15 years from now, there'll be 2 or 3 platforms that people will use. We're developing our own ways of doing this here, and we've got some really, really good people right at the cutting edge of the cutting edge, learning and pushing how to do this here. But there are a number of commercial platforms available, all of which basically provide some way of testing a patient's DNA and giving you a result that tells you some pharmacogenetic characteristic about that DNA. Hopefully it's something you asked for, but let me just give you 2 examples here. The Affymetrix DMET chip, and this stands for, I need Todd Scar, drug metabolism, elimination, and transport. Is that right? Yeah. But it has a couple of 100 genes on it and a larger number of genetic variants. And so you get a huge amount of data from using this approach. It costs about $600 commercially to run. Actually, the chip itself costs about that. It probably costs more to run, like Vicky. And that means that if I ordered this particular test, let's say for warfarin, I would get a lot more information than I needed just for warfarin. But of course, I've got a lot more information on many drugs that patient might be exposed to later on in life, other than just warfarin. So it might be useful for warfarin immediately, and it might give me more information than I would would ever need, but some patients want that, and some particularly vulnerable patients who are exposed to lots of drugs. I'm pausing for a reason to have you think about who those people might be. There are lots of patients who take many, many drugs and who are particularly vulnerable. As people get older, they take more medications. Over the age of 65 in the United States, that number is five prescription medications per day on average in the United States. And we know that at IU Health, in our systems, 92% of the costs are incurred by 7% of the people. And those people are on a lot of medications. Going down here, another example is autogenomics, and this should have a TM attached to it as well. It's a company, but this is a way of testing a very small number of genetic variants in a very automated way that gives you a very simple answer out at the end. So they're kind of these two extremes at this level, but of course there's also coming next generation sequencing, which will provide the entire, well, actually it's not, it's marketed as the entire sequence. It's not the entire sequence. There are bits that are hard to get, but most of the sequence, meaning more than 80%. And particularly in cancer, I think that latter approach is particularly important. But this is really just lots of possible ways of doing it. So one has to ask in the clinical setting, do you want tons of information, or do you want a small amount of information because you have a very, very narrow specific question? So here are some things to think about when you're making that decision. For simple tests, they're generally robust in dirty clinical environments. I don't necessarily mean physically dirty, although I am thinking about some physically dirty clinical environments. But I mean complicated clinical environments where you're very busy emergency rooms, very busy ICUs, very busy hospital environments where lots of things are going on and information is dirty, if you like. And if you throw a huge amount of information in the form of a next generation sequence into that environment, it's very hard for it to be effectively interpreted and used. But simple tests are robust in that kind of environment, and the number of screw-ups you can make is less the simpler the test. And that's something I think that's really, really, really important as we go into this now, because I think the first tests that we're going to have are going to be simple ones, not complicated ones. They're simple because they're simple to do. They don't take 16 people to operate or perform, and they're simple because they're simple to interpret. So literally the person who ordered it, or even the patient, ought to be able to understand how to interpret it. And I don't think that's generally the case right now. The implementation of the therapeutic change and approach is clear. So what you have to do as a result of the test is clear. And from a hospital system point of view, simple tests can be easily monitored for quality control. It's really difficult to do quality control, even on something that has 50, 60, 70 tests at once. If I have to monitor one test, like a CBC, complete blood count that's done every day in a lab, that's pretty easy to do. But if you've got something that's got 60, 70, more than that, variants on it, that gets progressively harder to do. And if you stick in numbers like 1 million plus, you get a very complicated quality control issue. And of course, these simple tests miss the possibility of multiple genetic contributors. We know this. We know there are genetic things we can't see, and the simple tests will miss them. So they're not as powerful predictors as we could probably generate. But if we're honest about it, there are none of these tests yet cover the complete genetic background. We haven't got all the genetic data we might need. On the other hand, the arrays are more inclusive. They're not completely inclusive either, because as I mentioned, even if you do a couple of million variants, you aren't getting to 3 billion, which is the total. You're not covering the whole thing. There's less need for platform adjustment, and I think this might be, from a business and economic sense, ultimately important. So we don't need to change the platforms as we move forward. We need to switch it to a different machine or switch our interpretation to a different method. It's one thing, and we just follow that. And obviously, they have greater flexibility of content. That's very important to us as we go forward in this enterprise at IU Health, because we anticipate we're going to need to add tests and subtract tests as things go forward. So for all of you, I think we have a pharmacogenomic platform that will begin, and we anticipate that it will grow with time. we'll add things and subtract things as the science develops. And ultimately, what we're looking for is validity. So I'm going to give a quick primer on what we mean by validity. And this would work really for any laboratory test, but it's something far too ignored and that not enough of us are familiar with in the world of reimbursement. So the reimbursement guys want this kind of information. And before I get into reimbursement, I want to just say that it is what's most surprising to me about the reimbursement environment, I think, for these tests is how much is reimbursed. Not how little is reimbursed, but how much is reimbursed. The majority of pharmacogenetic tests ordered at this institution, in fact, every one that I've ever ordered in my clinic has been 100% reimbursed. Now, that's not universally the case, and it probably, as this gets bigger and the big insurance companies notice it more, that's probably going to be less the case, but it seems to be running largely below the radar right now. It hasn't been hit badly. But the reimbursement guys want to know these four things. They want to know about analytical validity, which is simply that it's reproducible day to day in a lab. They want to know about clinical validity. So that's the extent to which a test accurately predicts the outcome. Now, you can have a test that accurately predicts the outcome, but is clinically useless. So I'll try and make this clear. For example, cytochrome P452D6 that Brian mentioned is it's a genetically polymorphic enzyme. It's absent in 7% of the Caucasian population completely, and it accurately predicts the concentrations of the active metabolite of tamoxifen. And this is a finding that we made ourselves more than 10 years ago now, but this does not associate with clinical outcomes in breast cancer consistently because there's more things involved than simply the concentration of the active metabolite. There's lots of things involved here. So this is a case of clinical validity that doesn't have real clinical utility. Clinical utility, on the other hand, I'm sorry, this is just me. Brian used this slide already, so I'm going to skip past it in the interest of time. Clinical utility, on the other hand, is where you have reliable evidence, and this is where they want to reimburse and where, if you like, the lawyers are more interested because if we don't do this, then we're more vulnerable. Where there's reliable evidence that the genetic variant is consistently associated with a clinical outcome, and here come the keywords, that alters or practice or is associated with improved patient outcomes, alters our practice or is associated with improved patient outcomes. So if you have a test that means you would do something, change a drug, change a dose, or monitor more closely, those three things, and I guess the fourth is stop the drug completely, then that's something regarded as having clinical utility. Something that has clinical utility, it's just kind of like speaking French, but if it has clinical utility, those words mean it's much, much more likely to be reimbursed, and it's much more likely to have a guideline attached to it, a CPIC guideline, and it's much more likely to be widely recognized by the practice community. Here's an example of that, just to see how long this takes or how fast it is, if you like. So this is time along the bottom and the number of test orders for HLA-B5701, which is the screening test for abacavir hypersensitivity. And I haven't put in the nasty slides of people's skin desquamating, which is the reaction that occurs if you prescribe abacavir to someone who has this variant. So what we want is to give abacavir, which remains a first-line therapy in HIV, to patients who don't have this variant. But here's the first association between the genetic variant and the skin reaction, published in the first quarter of 2002. Okay, 3 years later, the first quarter of 2005, the first clinical test is introduced. That's actually quite fast. We got Ken Levy here who knows how fast that is compared with many other situations. And then you go, the scale changes a bit here, but this is all 2005, 2006, nothing really happening in terms of the use of the test. So this is a clinical test is available, but it's not being used a lot. This line is still at the bottom here. And then it starts to take off around the time of the association being demonstrated in AIDS patients at the International AIDS meeting in South Africa in 2007. And then shortly after that, an HIV guideline comes out of the NIH, which is basically generated from the presentations made at the International AIDS meeting. And then this stuff presented in abstract form here in the second, third quarter of 2007 appears as a New England Journal publication in the first quarter of 2008. So we're 6 years after this, and there's a New England Journal publication, and we're way up here. Now it's being widely used. And in this case, the FDA comes in here last to the party. Everybody's doing it already, it's widely used, and the FDA finally says, okay, you guys can use it. This isn't the case every time. In the case of warfarin and clopidogrel, a lot of people would say that the FDA did this too early. Came in and said you should be using it when there wasn't sufficient evidence. So in some parts of the FDA, which unfortunately is a very large organization, 10,000 people, right, are very proactive and ahead of the ball, and other parts of the FDA are just behind the 8 ball. And they were, I think here, they finally did it, but they were slow. I guess an important message from this slide is that there are many, many, many tests on the clinical market. In fact, the majority of tests that we use in clinical practice are not FDA approved. Are not FDA approved. They're not, they will never be. There's no chance it's ever going to happen. FDA approval for clinical laboratory tests is a controversial subject that we have some experts here. But I think the majority of tests we use in clinical practice, things like Chem7s and Chem24s and CBCs and CRPs and things like that, are not FDA approved tests. They're tests approved by an organization called CLIA, which is the Clinical Laboratory Improvement Amendment. But FDA approval is not something we require for most tests. It is approved, though. It is provided for some pharmacogenetic tests. We'll get into that. At last, after clinical utility, we need this point about economic utility. The evidence that the use of a test results in more effective care as measured by decreased costs or improved outcomes. And this has been studied with the warfarin pharmacogenetic intervention, but probably not studied enough. And we really don't know in our system, as we start to do this in the next few months, whether this is going to decrease costs and improve outcomes. But for the first time, I think Indiana is really going to be one of the first places that this is done, where we look really carefully to see for our system, for IU Health and for Ashkenazi and Wishart, does the implementation of this technology really decrease costs to the system, and does it make us really better at what we're doing as measured by cost, which in many ways is the most rigorous outcome. Just to give you a sense of cost in a specific example that Brian mentioned, this is a 2C9 per metabolizer, so it could be someone similar to the case that Dr. Decker described. So this is a person where you're looking at the dose of warfarin on the y-axis, which normally is 5 milligrams. We start people at 5 milligrams of warfarin. And whenever I'm doing that, I tell them that I don't know what the ultimate dose is going to be. But you can see that the ultimate dose in this patient here, this is the triangles down here, are the dose. The ultimate dose here is actually 0.5 milligrams at some irregular interval of days. 0.5 milligrams every 2-3 days, as opposed to 5 milligrams per day, which is the normal. So that's more than a log lower than you normally give. And the point I always make about this, and every physician knows, is that if you overdose warfarin, as was done by accident early on here, you can cause cerebral bleeds, and then you're basically in prayer mode. There's nothing much you can do at that point other than ventilator care. So here is the INR, the International Normalized Ratio, after the 5 milligrams, which is hidden in here, boom, way up to a scurry level. You want it in here between 2 and 3 in this particular patient. So in this patient, the dose is reduced from 5 to 2.5 to 0 because it went so high, and then started again at 1, and it still went too high here, and then decreased to 0.5, and it still went too high here, and then they went down low, and it dropped below. So you can see, imagine how many visits this is to the doctor, how many blood draws, how much traveling for the patient, how much to and fro on the informatics system. It's a very costly enterprise as opposed to getting this right on the first shot. So tools going forward for rational prescribing, and this is the last couple of slides, and I'll take some questions. We would like to have pharmacogenetics included, and we're going to do that in our system to try and use it as much as possible. Ultimately, I think we'll use other things as well, but we're always going to do this in the context of these other normal things: history, age, ethnicity, renal, and hepatic function, in the normal way of doing rational prescribing. So I think pharmacogenetics here has a big future. We're going to try hard to implement it, and we're going to try hard to study it while we're implementing it to see if it's saving money or not. And I guess this whole meeting is a way of pointing out the fact and making clear to all of you that we're going to have a lot more people involved in doing this than we have up until now. Where are we going? I think we're going to robust, evidence-based, and reimbursable tests that do save costs. We'll know the answers to some of these questions. We're also going to test, and I think this is important, that combine clinical and genomic scoring algorithms. So we'll end up with scores and really clear recommendations for clinicians. And I'm saying that word carefully because I think pharmacists and nurses are going to be easily as important as physicians in this. But they'll combine the clinical assessments with the genomic assessments. So we have a combined, more powerful tool. And third, we'll have a lot more people who can do this. Healthcare professionals trained in using these tests to improve effects, decrease side effects, and save costs. And I will stop there.